Thank you very much. Thank you. So before I start, I would request, most of you could mute the microphone, because I know they have sensor detection. Sometimes they will switch to the people who are talking. So I'll start share my slide from the beginning. So so I just test a couple. Yeah, it works. Okay, so we all know this COVID-19 global pandemic. It's extremely challenging situation. It's causing lots of damage. The major cities are locked down. Globes. We know people lose their lives every day. I hope we can all work together so we can get through this adversity. I wish the best health, prosperity, and success of the McGill students, faculty, and staff. So the talk, before I start, lots of people ask me questions. Being a biologist, what's the difference between a biologist and between a chemist? I want to ask you one question. What's the difference between the chemistry in a living cell and in a test tube? So from my perspective, a single cell contains hierarchically organized information. For example, a cell can evolve, can adapt, respond, and maximize the cell's benefits. We call it biointelligence. So my lab basically focuses at the interface between synthetic biology, metabolic engineering, and intelligent control. So we are aiming to discover the metabolic cost, evolutionary stability, and social interaction of the cell at genetic, biochemical, and multiple species scale. We basically engineer genetic metabolic circuits that achieve adaptive biological function. We also construct microbial cell factories for energy, environmental, and bioproduction of biomedical application. So the second slide is actually this area start with the biomanufacturing. We know from thousands of years, almost all the food, drinks, drugs, vaccine we use on a daily basis are derived from biotechnology. Even today, 20 to 30% of the fuels, the chemicals, the materials also come from biotechnology. So OECD, which is the Organization for Economic Cooperation and Development, predicts around 35% of chemicals will be produced by biotechnology in 2030. So the benefits, we could use a variety of renewable feedstock. So we can reduce our carbon footprint, CO2 emission or greenhouse, so we can save our Earth. The biotransformation chemistry is very specific and environmentally friendly. We could make lots of commodity chemical nutrients, drugs, and fuels. So this is all about the start of this year. But the more interesting is that if we look inside of the cell, look about the biochemistry, the genetics. So let's first take a rhetorical question, rhetorical example here. So bacteria or yeast can be viewed as a cell factory. You have carbon goes in, you have product goes out. Inside the cell, you have interconnected, interconvertible metabolic network. So each of the edge in this network represents a metabolic reaction or chemical reaction catalyzed by a specific enzyme. We call, each of the mode, each of the node or the dots, we call it a chemical. We call it metabolites. Just like a chemical plant, we have feedstock processing unit, so which is the glycolysis in the cell, which is the glycolysis or the pentose phosphate pathway. We also have our powerhouse. Mitochondria generates ATP for cell growth and protein synthesis. We also have the assembly line. The cell makes functional polymers by assembling small pieces of chemicals. For example, proteins are assembled from amino acids. Cell membranes are assembled from phospholipid. We also have the hydrogen plant, which provides very important reducing power for repairing DNA damage or maybe scavenging the toxic chemicals. So how does the synthetic biologist visualize the cell? So the DNA or the genome basically is a blueprint for biological activity. A chemical engineer, they can write machine language, they can use central computer. We can just code with C+ language, maybe Visual Basic or maybe C+ or Python. We can control the mass flow and the energy flow in the chemical plant. But in the biology, actually, as a biologist, a synthetic biologist instead rewrite the genetic software, ATGC. We can control the mass flow and information flow in a living organism. By doing so, we can actually digitalize and reprogram cells for energy bioproduction and biomedical applications. So the next slide. So in my lab, actually, we are focusing on a very interesting yeast we call the YIN lipolytica. It naturally makes lipids up to 20% of the cell biomass. This yeast grows very fast and can use a broad range of substrates, including hydrophobic compounds, toxic chemicals. It's genetically tractable. We can engineer them. So the cell contains lots of acetyl-CoA, malonyl-CoA, HMG-CoA, which are the precursor molecule for lots of the natural products and pharmaceuticals. So the lipid biosynthetic pathway is well characterized in this yeast. There's one people joining. I'll just admit the people. Michael, okay. Welcome, Michael. So the lipid biosynthetic pathway is well characterized here. Because of lots of the lipid it can make, this yeast has abundant internal membrane structures. And the organelles, including ER, endoplasmic reticulum, peroxisome, lipid droplets, and hydrophobic compartments. So this oil-water interface actually creates the perfect environment for lots of the plant enzymes, which catalyze the P450-mediated hydroxylation, epoxidation reaction. For example, the cytochrome P450 enzyme, which with the most diversified function in nature, makes lots of the expensive antiviral, anti-tumor natural products. A summary here. So the internal membrane structure in this yeast actually just 1 minute, okay. The internal membrane structure in the yeast, I'll try to. The internal membrane structure, there's one more people joining. I'll just, okay. So they provide the possibility which allows us to explore the complex sterol radioselectivity of many enzymes. For example, shown here is the P450 enzyme diversification chemistry. From a single core molecule called beta-amyrin, we can get of thousands of different diversified molecules with different biological functions as antiviral, anti-tumor activity. So today, I'm going to share with you a few concepts about synthetic biology tool development in my lab, which can speed up and streamline our engineering process, and the yin-yang balance concept, which helps us to engineer efficient microbial cell factor. I'll also give you some example how we can engineer precisely control the biological function of genetic circuits to control carbon flux distribution and eliminate metabolic instability in this yeast. As an engineer, I'm also interested in, okay, there's two people waiting here. I'll just let them in, okay? Just 1 minute.
Peng, do not worry about the people. So that will be taken care of. You concentrate on the presentation.
Okay, sure. Because I see there's two more people on the waiting list. Thank you.
Do not worry about it.
Sure, okay. So as a chemical engineer, I'm also interested in developing mathematical models to understand biomolecular interaction at multiple dimensions, including genetic, cellular, and multiple species scale. So at the end, I'm also going to share with you some of the future research projects. So the first important genetic tool we built in my lab is the genome editing tool. We all know CRISPR-Cas9. We first tested CRISPR-Cas9, but it turns out there's lots of non-specific cutting, and the genome editing efficiency of Cas9 is very low. So part of the reason that Cas9 needs a tracer RNA, which is a trans-activating CRISPR RNA, the folding and the maturation of this tracer RNA makes the functional expression of the GAD RNA extremely challenging. The other reason that Cas9 can only recognize the NGG PAM sequence, so which is a protospacer adjacent motif sequence. So the GAD RNA, so because Cas9 can only recognize the NGG PAM, it introduced lots of the non-specific cutting. The reason because the genome of our yeast contains around 65% of the GCs. Imagine Cas9 only recognize NGG. The chances are that Cas9 will introduce this randomly. They can misread and miscut. They can introduce lots of non-specific cutting on a high GC content organism. So different from Cas9, Cas12 we also call CPF1. They can overcome these two limitations. For example, CPF1, they use a much simplified CRISPR RNA. We call cRNA. So the CPF1, it doesn't need the trans-activating part of the RNA, which can increase the likelihood of the formation of the GAD RNA CPF1 complex. Another advantage is the PAM sequence. CPF1 recognized TTTM motif, which has a very limited chance to mismatch or misread a high GC content organism. So we test, we engineer. So we set off to test the CPF1 genome editing in our yeast. The first thing we do is that we look at variety for promoters, which is the DNA sequence that allows the binding of RNA polymers, like starts transcription. We use a number of molecular mechanisms to increase transcription, in the processing and the folding of the guide RNA, including the use of self-cleavage ribosomes, hammerhead ribosome, and HDV ribosome, and the use of transfer RNA or the ribosome RNA promoters. We found that because these are the most abundant RNA, the transfer RNA, ribosome RNA are the most abundant in the cell. To our surprise, we found that the single type 2 RNA polymers was sufficient to drive expression of the guide RNA. Example shown here is the genome editing of the CN1 marker, GA marker, which encodes arginine permeate that allows the cell to take in the important amino acid arginine. So canovanin is structurally analog to arginine. In the agar plates containing canovanin, normal cells will mistakenly incorporate canovanin and will die. But the successfully genome edited cell will survive because they lose the ability to uptake canovanin in the middle. So the CN1 mutant cells still have their own kind of arginine biosynthetic capacity. That's why they can survive. Based on the canovanin resistant phenotype, we sequence the genome, we sequence the gene set. We found that indeed we found nucleotide insertion or deletion around the PAM sites, which demonstrate the success of the genome editing. Similarly, we can use a different genetic marker with UR3. We found very high genome editing efficiency, whether we target on the top strand or the bottom strand. We know DNA are double-stranded DNA. You can design the target sequence on either strand of DNA, on the sense strand or the non-sense strand. So the other genetic marker we look are the sulfur housekeeping genes. So methionine is a sulfur-containing amino acid. The biosynthetic pathway of methionine involved a number of sequential reduction reactions from sulfate to sulfite. For example, the SO4 managed to go to the S-2. Maybe I can click there's a pointer actually is function. I'll try to laser pointer. So you see here, so the SO4 managed to sulfate eventually reduced to the S-2 sulfide. So what happens that if we block the downstream step, MET25 gene, if we delete them, we can mutate them, the cell will start building or making lots of sulfide. The S-2 sulfide will be precipitated and form black color if we feed the cell with insoluble lead, such as lead nitrate, lead acetate. So that's exactly how we do in the lab. Based on use our naked eye, we can screen MET25 mutants. We found that this black colony, after DNA sequencing, we found the mutation rate could reach 45 to 75%. You know, for 3 different sulfur housekeeping genes here. My students are very creative here. They use the mutant cell and the normal cell. They made this yin and yang arts. We also found these MET25 mutants. Actually, they are very sensitive to detect soluble lead, up to 10 ppm. This could be a portable, inexpensive whole cell sensor for toxic chemicals, including mercury, arsenic, and cadmium, which cause lots of the health problems in developing countries. We also achieved the multiplex genome editing with efficiency up to 80% for 2 genes and 42% for 3 genes here. So based on yeast genetics, we have built a number of tools, including modular DNA cloning, repeatable and multiple copy chromosome gene integration technique. I would like to highlight one feature of this yeast. It can tolerate a very high amount of oxidative stress, or hydrogen peroxide. Data shown here, we have engineered a transcriptional factor-based biosensor as the first, from the data at the first 10 minutes, engineered yeast, they can carrying the sensor, they can linearly and rapidly respond to hydrogen peroxide. So around 10 minutes, the gene expression reaches the plateau and start declining after 10 minutes. This tells the transcription response time for yeast around 10 minutes. So the declining phase is because the cell has a defense mechanism that can effectively remove the hydrogen peroxide. So evolution is very amazing, it's powerful. This yeast makes lots of lipids. We know lipids can be easily oxidized to generate reactive oxygen species. This yeast had used two proteins called SKN7 and YAP1. SKN7 and YAP1 here. So initially, these two proteins are in the cytosol, but upon oxidation, these two proteins, they can form a protein complex under oxidative stress. This protein complex is later translocated into the nucleus and search for the specific DNA binding site on the genome, which activates the downstream gene that is responsible for getting rid of the reactive oxygen species. So this activation mechanism actually can be faithfully recorded and harnessed by our reporter gene. So we can engineer this genetically encoded biosensor, which can tell us the exact redox states of the cell, which may have lots of other biomedical or bioproduction applications. So the second part, I will illustrate a basic principle how we implement yin-yang balance to improve oleochemical production in our yeast. So what is the yin and yang in cell metabolism? So yang basically represents the carbon, including acetyl-CoA, malonyl-CoA, HMG-CoA. They contain the oxidized carbons. They have ketone groups, hydroxy groups, and the eno groups, which are electrophilic. They lack electrons. So yin basically represents the hydrogen, including the NADH and the NADPH. They are nucleophilic. We know the final destination for NADH is the oxidative phosphorylation to generate ATP in the mitochondria. All the biosynthetic pathways need the reducing equivalent NADH or NADPH to happen. Just take the example of the fatty acid elongation pathway. We need 4 critical steps. First, beta ketosynthesis, they can form the yang beta ketosynthesis, they form the yang molecule, another ketosynthesis, ketones. Then you need the NADPH to reduce the ketone group here. You have the beta ketosynthesis, you can form another ketone group. Then you need the ACP reductase. You can use the yin molecule to reduce the yang, so you can form a hydroxy molecule. The next step, you can use the dehydration step. You can remove 1 water molecule. You can form this eno group double bonds. Then use another eno reductase. You can reduce the double bond. You can form a saturated carbon with 2 carbon elongated. So what happens here, with each of the cycle running, we have 2 carbons incorporated. So the cell can keep running this elongation cycle. We can synthesize longer chain oleochemical like palmitic acid, stearic acid, even EPA, DHA for nutrients and some advanced biofuels. In our yeast, we found this yeast yields pentose phosphate pathway provide most of the NADPH. But this is not an efficient route to generate NADPH because there's one step is the decarboxylation. For example, we found that with each of the CO2 loss in the pathway, we only have 2 NADP generated in this pathway. So this will significantly limit the yield and the potential of this pathway. So based on the stoichiometric calculation, we next determine that the NADP actually is the real limiting factor to synthesize oleochemical and fatty acid. For example, here in the C18 stearic acid here, we need 16 molars of the NADP to running this cycle, which requires the cell to oxidize another 1.33 molar of the glucose. So not to mention, we need another 4.5 molar of glucose to provide all the carbon backbones. So the overall stoichiometry actually tells us we need, the overall stoichiometry tells us we need 5.83 molar of the glucose to synthesize 1 mole of the stearic acid. So the carbon conversion yield is very low. Eventually, from 1 gram glucose, we can only get around 0.271 gram of the stearic acid. So, the source of NADP actually determines the carbon conversion efficiency. Imagine the carbon, we cannot stay with the carbon because there's no easy way to do the carbon fixation or the condensation reaction in the chemistry, but the source of NADP actually can be easily managed in the cell. But the carbon consumption actually is directly linked with the cost efficiency. From this cost efficiency curve, shown here on the right, the blue and the red dots so the blue and the red dots are the carbon conversion efficiency. Is the fatty acid yield, and the golden curve actually the feedstock cost. The golden curve here is the feedstock cost. What we found here is that in nature, there are lots of alternative NADPH pathways, which tells us we can engineer the yeast and steadily improve the carbon conversion yield and lower the cost. For example, number 0 points represent the limits of the pentose phosphate pathway, which is 0.271 gram per gram. So we already calculated the number in the previous slides. We can further move the operational point to number 2, number 3, and number 1, number 2, and number 3. We can keep pushing the carbon conversion limits. I'll tell you in a second what are those alternative pathways. So next we have assessed and engineered 4 pathways corresponding to the operational point number 1, 2, and 3. So pathway number 0 is the pentose phosphate pathway. Number 1 is the NADPH dependent gap dehydrogenase pathway, and number 2 are the pyruvate oxaloacetate malic cycle. And number 3 is the non-oxidative glycolysis pathway. We engineered all the pathways. We identified a number of strains with carbon conversion efficiency reaching, right now reaching 0.286 gram, with beyond the limits of the pentose phosphate pathway. This is the first report we can engineer the yeast. It can beyond the native host. Our final engineered strain actually produced around 100 gram per liter lipid in a 3-liter benchtop bioreactors with a productivity around 1.2 gram per liter per hour. So this is truly remarkable because it allowed us to move us one step closer to the commercial or industrial application. So can we further push the envelope? We know mitochondria provide most of the NADPH, but the mitochondrial membrane is not permeable to the NADPH because of the reverse proton gradient. We know there's a ATP pump, there's a high proton concentration outside in the outer membrane of the mitochondria. To get this to work, we need to move around this we can move around metabolites across mitochondrial membrane. We found that there are lots of transporter protein. Actually, they can specifically transport dicarboxylic or tricarboxylic acid across mitochondrial membrane. Carboxylic acid actually can be a carrier or vehicle for this electron. For example, here I show here we can use acetyl-CoA as an electron to transport mitochondrial NADH. What happens is that in the mitochondria, we have an enzyme. It can be reversible. For example, there is a alpha-Kg dehydrogenase, which they can do a reversed carbon condensation reaction, use the NADH, ATP. They can form from the 5 carbon alpha-Kg to this 6 carbon NADH, so can deposit the NADPH reducing product. If you have a transporter protein, so transporter can transport acetyl-CoA to the cytosol. Then we can have a NADPH specific acetyl dehydrogenase, so it can oxidize acetyl-CoA, so we can give a reduced equivalent NADPH, so we can feed to the fatty acid biosynthetic pathway. And alpha-Kg eventually can go back to the mitochondria. So basically, we can transport this across mitochondrial membrane, but the essential thing here is that we use carboxylic acid metabolized as a carrier, as a vehicle. So this way, actually, we do the yin-yang balance, yin-yang conversion, because the carboxylic acid is a yang and NADH is a yin. So yang can be temporarily carry the yin. You can move the metabolites outside the mitochondria, so they can go to the cytosol, so it can provide the yin for the next reaction. So the next part I want to demonstrate, we can use this yeast to make polyketides, isoprenoid-based natural product with a high-value compounds. So this feature actually we take advantage of the malonyl-CoA, HMG-CoA flux, and the P450 expression platform this yeast. So flavonoids, why are interested in flavonoids? Flavonoids actually they are nutraceuticals, they are antioxidants, and they have cardiovascular protection. They can slow down the aging process, are very good to protect our neurological system. people have been demonstrated they are effective to, for Alzheimer's disease, Parkinson's disease, memory, they can do the memory reinforcement. Also, they are the very good signaling molecule to maintain the gut microbial homeostasis. People have been found they are very chemically diversified. There's around more than 10,000 analogs. Because of the side chain, you can do lots of hydroxylation or maybe prenylation, glycosylation. This has a lot of distinct biological function. So we argue that because the yeast, they can have a spatially organized organelle, including the ER, peroxisome, oleosome, which is the lipid body, they can provide the ideal microenvironments for the P450, VGO, or sterol selectivity. So we test this expression platform. We optimize the flavonoid and hydroxylated flavonoids production, which relies on P450 expression. We build the analytical. We use the HPLC to detect the metabolites. We have a standard. We have our standard hexfolin, eridictyl, and naringenin. We also have our sample. Once we express, our control strain actually only produce one of the naringenin, our initial strain. Once we engineer some of the P450 enzyme, flavonol 3 hydroxylase and the CPR pairs, we found we can produce both the hydroxylated hexfolin and eridictyl. We also optimize gene copy number. I will just keep the detail. We select a range of enzymes from different plants. We found actually the gene dosage has a major impact on the naringenin production. The next step, we found you need the best combination of the flavonol 3 hydroxylase and the CPR pairs. We did the bioconversion model efficiency, the conversion efficiency. Next, actually, we optimize the precursor pathway. We know the hexfolin and naringenin are synthesized from the phenylalanine tyrosine pathway. Optimize chemical acid pathway. Eventually, we found in the cell, we can engineer, we do some process optimization, control the pH. In the shaker flux, right now, we can produce around 250 milligram per liter of naringenin. This compound and around 120 milligram per liter of the eridictyl. This compound is very expensive. It could up to $200 per kilogram, or maybe sometimes $1,200 per kilogram, depending on the source and the purity of the compounds. So we found a number of problems in our yeast because we know all the flavonoids pathway, actually the naringenin and the flavonoid, naringenin, eridictyl, they all synthesized come from the common precursor called acetyl-CoA and malonyl-CoA. But in our yeast, it's a yeast we know is very famous to make lots of the fatty acids. Basically, the fatty acids and the naringenin, they compete for the same precursor molecule. So how can we smartly, intelligently allocate the carbon flux to make the cell has well enough amount of fatty acids to can maintain the cell growth, because fatty acid is essential for membrane lipid synthesis. At the same time, we can maximize the carbon flow, go to the naringenin pathway. The other one, we found our yeast is not stable. After a few generations, 10 or 20 generations, we found this yeast, they start losing the naringenin production phenotype because this is the evolution. So to solve these two challenges, actually we will take two steps. First, we try to engineer this negative autoregulation network. So we can engineer, so this layer one, this gray box area, we can engineer an inverter gate to can redirect malonyl-CoA flux to naringenin. What happens there, we can, in the yeast, they have a sensor protein called POR1P. If the cell know they have enough of the fatty acids, so the POR1P will be activated, so we can drive the expression of the GAD RNA. We know the GAD RNA, they can direct the activity of D-casein. D-casein basically is the catalytically dead casein. So we can, once they form a complex, they will repress the expression of the fatty acid synthesis pathway. So this is kind of a, autonomous regulation. The cell will have just enough amount of fatty acid. If they have produced too much of fatty acid, it will just automatically shut down fatty acid because there's a sensor regulator system here. On the other hand, to solve the stability issue, we can engineer this addiction phenotype. So addiction means basically the cell become addicted to this naringenin. So what happens that there's in plant, in bacteria, we found there's a heterologous protein called FDR. This FDR, it can be specifically activated by the naringenin. Once the protein bound to naringenin, they will become an activated form. They can activate the expression of the downstream gene. So in this yeast, we can delete the leucine biosynthetic pathway, LEU2 gene on the chromosome. We can place the LEU2 gene only under the control of the naringenin inducible promoter, which means we can build a strong correlation between naringenin production level and the cell growth, because leucine is the essential amino acid for cell growth. So in this way, actually, we can selectively enrich the cell which can produce more naringenin. At the same time, the cell can autonomously adjust the carbon flux. It can maximize the carbon flow go to naringenin. So we did lots of experiments. It's all concentrated on one slide here. For example, here, we screen the fatty acid responsive promoter regulator system. We found this hybrid promoter PA1, R1A3 promoter, and PAR1P pair. We also engineered this flavanol reducible promoter. Indeed, we found naringenin level can dictate and dominate cell growth fitness. In the agar place, if we in the absence or without supplementation of naringenin, the cell didn't grow. But in the presence of naringenin, we found abundant cell growth. In the liquid media, we do test the cell display a very distinct growth tab, growth phenotype. With naringenin, we found cell growth much faster. Without naringenin, the cell grows very slow. So we can build the correlation between the cell fitness and the naringenin production. In the end, we found, we test cell growth fitness. Almost our engineered cell and the control cell, our control cell just, with the naringenin production gene without the layer 1, layer 2 control circuits. We found our engineered cell actually, they start gradually increasing their growth fitness, but the production actually decreasing a lot after 200 hours, still in inoculation time. But for engineered cell, we found our cell can persist around 91% naringenin production up to 300 generations, up to, normally it's around 2 hours per generation, 600 hours inoculation time corresponds to 300 generation. So the cells without flavonoid addiction basically regain the growth fitness, but they lost around 95% naringenin titer after 300 generation. Here in this control cell, they lost around 95%. So, so the example shown here, we can use intelligent control. We can use combine evolution and the engineering principle. We can improve the stability. Imagine you grow this cell in the bioreactor. So you don't need to worry about the genetic stability, genetic instability on the strain degeneration. So another example here, we can engineer the isoprenoid amorphidine. We know malaria is a life-threatening disease and causes more than 200 million infections, around 500,000 deaths in 2016. Most of them are women and kids in African sub-Saharan area. So the most effective drug is the ACT called artemisinin combination therapy with quinine and artemisinin. But the artemisinin global demand is very large, around 250 to 400 metric tons. But the current plant extraction, due to the low yield and the long cultivation cycle, normally need 9 months in a very hard area. They cannot provide a sustainable supply chain of the low-cost artemisinin. That's why our group was founded by the Bill Melinda Gates Foundation. So we found actually when we try to engineer, we move around here. We can move around the metabolites from the peroxisome. We found the peroxisome actually a very good storage space. It can provide acetyl-CoA. Eventually, we move around the metabolite. We overcome the transportation limitation in the cell, also the NADP supply. Eventually, we can engineer a yeast that right now can produce around 180 milligram per liter of the Artemis amorphidine, which is a precursor. It's for amorphidine, there's only a couple steps away from artemisinin. Based on the same pathway, actually we can engineer a squealing. So the squealing basically is we have the structure of this one. They call it shark liver oil. It's a thermostable super lubricant. It has the skin compatibility. It can be used in the cosmetic industry. So as a precursor for lots of steroids, for example, the testosterone, some hormone-based drugs. Also, it's the first important precursor for triterpenoid biosynthesis. Just with the expression of a couple of genes, we found our yeast it can produce around more than 500 milligram, half gram per liter of squealing in the shake flask. If we can scale up in the bioreactor, I can imagine it can increase to 10 gram per liter, which will become very promising. So we don't need to kill the shark. We don't need to go to the ocean to kill the shark. So in the last part, I want to I want to demonstrate we can use biophysical models to understand genetic circuit dynamics, design principles, and metabolic network constraints and microbial consortium interactions. A couple years ago, my lab has engineered a malonyl-CoA sensor. We found actually the sensor structure is quite interesting. We built this because we have a T7 promoter. We just use lipolytica and T7 promoter. At that time, we don't know too much of the synthetic biology. We just place the 5-PAR, which is the malonyl-CoA responsive protein, under the control of T7 promoter. Also, we place the reporter gene under the control of this hybrid promoter. In our experiments, we found actually as a tuning point, we can use IPTG to remove the active lipolytica, which is the repressor. They can repress the T7 promoter. So as we increase the IPTG, what we expect to see that we should see increased gene expression. What we should see, this is our expected. But in reality, our experiments result, we found as we increase the IPTG, as we increase IPT from this is the lowest concentration to the highest concentration here. So this is a control string. So the lowest concentration is for example here. So as we increase concentration of IPTG, we found actually, so this is the blue dots here. So this one, the blue dots here is the highest concentration of IPTG, and here the pink one and the red one is the lowest concentration of IPTG. As we increase concentration of IPTG here, at the first phase around 300 minutes, we did find our experiments. Actually, we found they follow exactly the trend, our expectation. We found that at the first 300 minutes, we increase the IPTG. But as we go through, after 300 minutes, we found they reach the plateau, it started declining. The reason because there's a crosstalk. If you just look this line, I just tune down okay. If you just look this line here, you found basically the lipolytica actually they repress the GFP. That's why the IPTG they can remove the lipolytica activity. So IPTG will induce the expression of GFP. But after 300 minutes, the cell start making lots of 5-PAR protein. The 5-PAR protein start joining, they can repress GFP. That's why we have the second phase. There's enough of 5-PAR, they can start declining. The GFP expression will be declined. That's why we have this crosstalk. So another thing that we also try to engineer the growth, understand how the cell grows in a bioreactor. In the traditional approach, people use this, they call the monod equation for this part, which is the nutrient-dependent term. We also found as the cell become very crowded and the nutrients become limited, the cell start having self-inhibition effect. We call it growth inhibition term. So we incorporate both terms. Actually, we can analytically solve the solution in both the batch culture and the CSTR culture. So this equation represents both the limitation, the transition, physiological transition from the nutrient limiting condition to the growth inhibition condition, depending on the yield, the biomass yield. So we can become it's published on BNB in just the beginning of this year. So another challenge is that we can, we can look about the microbiome, human microbiome. People found there's around 100 more genetic diversity in human blood system than our genome. So because of the heterogeneous population, there's tens of thousands of species. You can call, so the dysbiosis or maybe the instability of the population that can cause the inflammation, metabolic disease, Crohn's disease, inflammatory bowel disease, or the neuron brain gut nexus. People found the kids autism, attention deficit hyperactivity disorder, or depression. It can be linked with the gut microbiome. So also they can cause the innate immune system. So based on the two species, A and B, based on the interaction, we can introduce an equation. For example, here, we can modify the growth fitness effect function. We can introduce this interaction coefficient. Then we can model the cell growth in the CSTR. We found we can identify at which social interaction they can co-op, they can stably coexist. We found in this case only the parallelism and this one cooperation, or they can do a coexisting in the bioreactor. For other cases, if they have a strong competition, there's only one species will exist in the final state. Only one species will win the competition. Other things, a couple years ago, we engineered a malonyl-CoA sensor. We found there's oscillation malonyl-CoA behavior. It's published on PNAS paper. At that time, we don't know exactly what happened. But after that, because I'm a chemical engineer, I'm interested to understand what happens behind in the cell. We come up with some mathematical equations. We can replicate the gene expression dynamic in the cell. So we can do a three-dimensional phase plane depending on the fatty acid, malonyl-CoA, and fatty acid synthesis. We found there's a multiple looping behavior, which means there's a multi-stability of the cell. So this metabolic switch basically, depending on the malonyl-CoA sensor, they can either upregulate and the upstream pathway, and they can repress the downstream pathway. This is the on-off control, achieve both on and off control in the cell. So in the summary, so we would say the genetic tools are very important, which helps us to streamline and accelerate pathway engineering in the yeast. And yin-yang balance is a very critical concept. So it can help us move around the electron, the carbon, so they can maximize the carbon conversion efficiency. And the Yarvia lipolytica is a very promising yeast to make natural products, including flavonoid, isoprenoid. Dynamic control actually, and the transcriptional reprogramming is the essential genetic tool which allows us to design and understand cell metabolism. In the next 3 to 5 minutes, I want to present you the major research project in my lab. So the first project, I want to upgrade the low-cost agricultural feedstocks to antiviral, antimalarial, or maybe the recreational drug through the yeast platform. The next project, I want to focus on the microbiome. I want to understand the microbial social interaction, dynamic population control, and application. In the end, as last project, I want to understand the biological signal processing process, engineer process to control the biological genetic metabolic circuits. We all know this yeast is very good. This is I just try to skip that. This yeast, because it can make lots of the acetyl-CoA, this is the starting point for lots of the commodity chemical, polyester, hydroxy fatty acid, polyester, omega hydroxy fatty acid, even short chain fatty acid, West esters. Also, they are very good to making the P450 enzyme expression, to make functionalization chemistry to diversify the natural product. So we can upgrade, here I just identified acetic acid, actually the very good source for our yeast, because our yeast can use lots of short chain fatty acid. Lots of the agricultural waste from anaerobic digestion, you can produce lots of acetic acid from acetogen fermentation process, even lignocellulose, agricultural waste. So we do the stoichiometric balance. We found actually acetic acid can save up to 33% of the efficient. They can save 33% of the carbons. So the first project I want to engineer this, butyric acid. which branched from the secreting biosynthetic pathway. You need a downstream a number of the P450 enzyme expression, which requires NADPH, because our yeast, it can tolerate lots of the oxidative stress. They have abundant NADPH. They also have lots of the precursor molecule. They can make lots of the secreting. So, for example, for other pathway, I'm interested in making the compound called quinine. Quinine, people have been achieved the chemical synthesis, the very complex steps, the yield very low. But the quinine actually naturally derived from the cinchona. It's a plant only found in Southeast Asia. Also, I want to engineer this glyceric acid, so which only found in the licorice. It's a traditional Chinese herb. So this has a very strong antiviral and HIV activity. Of course, other things, I want to engineer this recreational drug with this our yeast platform. We can use, because it can make lots of malonyl-CoA. We can feed the yeast with the hexanoic acid, hexanoic-CoA. Eventually, they can make oleochemicals acid. Combined with the perennial transferase and the GPP, which branched from the malonyl pathway, we can synthesize CBD, which is the starting molecule to make CBD and THC. We know CBD, it has entertainment and recreational drugs. It's licensed, legalized in Canada and lots of countries. And THC actually they have a psychoactive activity. So because the marijuana industry, people has been estimated by 2030, so the global market could reach to $20 billion. So this is a huge market. I think with the license going on, I think it's possible we can engineer this pathway in our lab. So the number of steps, I just don't try to skip it. Basically, we have a very solid planned steps. We try to overcome lots of limitations and diversify the P450 enzyme expression. For this marijuana pathway, they also have lots of P450 expression. So the third project, the second project, I want to understand gut microbiome. How do we, model the microbial social interaction? How do we apply dynamic population control? For example, if there is one or two species, if they share the same substrate, the two species can mutually exclude each other, or maybe they can mutually promote each other. They can also do a cross biotransformation from the metabolites, can cross transform to product P1, P2. Of course, the metabolites, they can crossly inhibit each other, or maybe they can promote each other. How do we define the social interaction? How do we identify the coexisting states? Even for a 3 species population, we may have 64 different social interactions. If you consider about mutation, evolution, genetic instability, it's more interesting phenomena. So we can understand probiotic pathway interaction in the human gut system. We can combine with the microfluidic professor in our department. We also had host microbial interaction, for example, the soil microbiome plant on the plant roots. For biomedical, we can understand the tumor and the normal tissue interaction, and you can evaluate genetic stability, antibiotic resistance. The last project, I want to use computational model and experimental tools to understand signal processing process in biology. For example, we can build this genetic metabolic circuits. For a single genetic metabolic circuit, for example, there's the DNA or the transcript or mRNA, which encode the enzyme. But the substrate could be repressed. There's a repressor protein. They can regulate repress expression of the original coding sequence of the DNA. But the metabolites actually can work as a co-repressor or activator for the repressor. The final product could be an activator for the repressor protein. So cell has a very elegant control system. They know exactly how much of the metabolites gene expression they want. For 2 enzymes, 3 metabolites, for example, M1, M2, M3, 3 metabolites, E1, E2, 2 enzymes, we may have 64 different genetic metabolic regulatory motifs. For example, the enzyme 1, enzyme 2 can be put under the control of the upstream or downstream promoter. Each of the promoter can be either upregulated or downregulated by a transcriptional factor. But this transcriptional factor activity can be controlled by the intermediate metabolite M2. Also, we have feedback regulation. M2 can be either feedback, inhibit, or activate this pathway, the upstream pathway, or maybe M2 can activate or repress the downstream pathway. So just like the electrical engineering, the ILC circuit, contains resistor, inductor, and the capacitor. They have you can build lots of the interesting, interesting signal processing process. You have a signal input, output, bandpass filter, low frequency, high frequency. In the biology, actually, the bonding process of the metabolized transcriptional factors, we can exactly model them. We can try to understand the transfer function. We can come up with genetic resistance or maybe conductivity or metabolic conductivity. Understand the time response dynamic, the transfer function of the cell. For example, here, use just a simple bonding kinetics. We can, with the fraction of the occupied bonding set, we can model the, the transfer function, the input output dose response curve. So this is just for a single scenario. For 64 scenario, this is just a very simple regulation. If we incorporate the metabolic regulation, it could become very complicated. So in the end, I would like to thank the search committee for inviting me for the research, for the seminar. Also, I want to thank our funding agency from National Science Foundation and Bill and Melinda Gates Foundation. Thank you for all your time, and I'll be happy to answer your questions. Thank you.
Thank you, Peng. The questions, it would be better if the questions are typed in the chat, but certainly the questions can be all right, so that was clapping hands. But the questions also can be asked in person, and sometimes it is needed. So sure. the presentation is open to questions now.
Thank you. So to ask a question, you first need to unmute yourself, okay? Make sure you first unmute yourself.
Look, we do know we use Zoom for quite some time. So, all right, yes. So we have a question from Robert.
Okay.
So you can read the question, and it could be good if you repeat the question and then answer the question.
Okay, wait. I need to check the, yeah. I'll just check the question here.
The chat.
Yeah, okay. Yeah, okay. So the chat, yeah.
From Robert, the second question.
Okay, I'll try to, yeah, from the chat. Wait, so the chat, okay. Yeah, I'll just go to the chat function. Okay, just one more minute. So yes, open it. So the question that, so what are your thoughts on whole cell simulation models, such as flux balance analysis? Are they good? Thank you, Brandon. Okay, so another question from Robert is, ILC models are spatially lumped. Is there a possible need for spatially distributed model? Yes, those are very good questions. So ILC model, we know there's a complex signal process, but in the cell, actually there's a concurrent parallel signal. At the transcriptional signal, mRNA level, at the metabolic signal, you have metabolite expression, DNA signal, you have a template DNA. So the cell actually is very complicated. They have compartment. So the spatially lumped, I do think at the present moment, I'll first approach this question with the homogeneous system. We just think the nucleus as a well-mixed system. But in the later stage, if we try to understand the differentiation of the signal, we need to introduce the transfer, problem, the coefficient diffusivity of the process. But at the first beginning point, I think we would just do think this is a homogeneous system. But we do want to come up with concurrent parallel signal processing. For example, we can build the filters, the low frequency bandpass filter, or maybe high pass band filter. So what's the equivalent, biological equivalent for capacitor? What's the biological equivalent for resistor? What's the biological resistance for inductor? We can first find the analog for the biological system. Then we can come up with what's the dynamic response. We can engineer them and build them and observe dynamic behavior. But in the beginning, we just engineer them in the cell-free system in the test tube. Well-mixed system, we think we don't need to consider the spatial distribution. But in the future, we do need the spatially distributed models. So the second question from Brandon, what are the thoughts on the whole cell simulation about flux balance? So flux balance, basically I worked with, Costa Marinas like 6, 7, 8 years ago at Penn State University. So we built some genome scale flux balance model. So this flux balance model are essentially based on the stoichiometric relationship. They are very good to predicting the cell fitness, growth rate, and the flux distribution. Also, they can predict the genetic overexpression targets. But one limitation that they didn't incorporate the regulation, the transcription regulation, or maybe the post-transcription regulation. So they just predict the static solution. They didn't consider the dynamic change of the cell system. That's why the whole cell simulation, it will be very complicated. Whole cell kinetic model. Right now, I don't think any computer can handle a whole cell dynamic model, but we can just take a snapshot, a part of the cell metabolism. For example, only the glycolysis pathway, or maybe only the lipid biosynthetic pathway contains 30 to 50 steps. We can understand the regulation dynamics. But in the future, with increasing our computational capacity, I think it's possible we can consider whole cell dynamic simulation models. Thank you, Brandon and Robert. Thank you.
Are there any other questions?
Okay.
Okay, Alan, yeah. Sure.
Sure. There's another 2 general questions on efficiency. First is, can you compare the efficiency of a biosynthesis production with the chemical process? So how much energy goes to living and building? The second question, what do you think are fundamental limits and obstacles for biosynthesis efficiency improvement? So this question, we know, yes, this from Professor Allen, and so this question is very important. I just have talked with lots of chemical engineering department. I have worked in the chemical engineering department a lot. For people working in the heterogeneous catalysis, working on the petrochemical, petroleum feedstock, they just argue with you, what's the benefit, efficiency the cell can achieve? So one thing that I just want to mention, the cell, actually, the chemistry are very specific. Sometimes the turnover rate may be slow, but we can do protein engineering. We can improve the catalytic efficiency. Also, we do not need lots of the toxic catalyst. We don't need a high energy input. We just need a green process, environmentally friendly process. For lots of the process, we can find a niche, where we can make it economically possible. There's around 200 startup companies. Every year there's 20, 30 startup companies in the East Coast, West Coast area. They all work on renewable chemicals, feedstocks. So for example, 1,3-propyl, which was invented by DuPont, actually it could be quite efficient. It's better than the petrochemical conversion process. For example, another one is, but most of the current products focused on the nutraceuticals, high value compound or drugs. But for the green chemical, I think we do, on the polymer industry, we do find there's some potential. We can improve the efficiency. Eventually, it boils down to the dollars per pound, the cost efficiency, how much of the resource you input to the system, how much of the final product you get from the system. So what's the fundamental limits and the obstacles? I think the fundamental limits are sometimes you have the thermodynamic barrier. For example, some of the enzyme reactions are non-reversible. You have to use alternative steps, try to bypass the step. Other thing, the cell, sometimes the growth, the fitness. On the process, the cell may grow, they lose their fitness, or maybe they become intolerant. There's all kinds of oxidative stress, or maybe chemical toxicity issue. We have to think about the process integration, come up with integrated process to recover, simultaneously recover the final product, and feed the precursor molecule and improve the transportation, transport efficiency, or maybe the biocatalytic efficiency. So the protein structure and the protein catalytic efficiency, also the growth fitness, the stability of the system, I think are the limits of the current biosynthetic efficiency. Thank you, Alan.
All right, so if there are no other questions, and in the interest of time, we need to move to the teaching presentation.
Okay, thank you. I'll just stop share. Okay, I'll just stop share. I'll just switch to, okay. I'll just switch to a different screen to share with my, okay, I'll share with the, I'll turn off this teaching, I'll start a new share to share with the, okay, I'll start, go to the teaching seminar, okay, from beginning. Okay, so today's topic is very interesting because as a bioengineer, we just want to solve some challenge. Actually, like a couple months ago, I submitted a paper. I tried to model the latent transmission dynamics for coronavirus, how it transmits from people in Wuhan, in China. Because this virus has a 1 week, 7 to 10 days latent phase. People catch the virus, they don't know, they just keep spreading the virus. That's why we have see so many people get infected. So as an engineer, I want to mention in this teaching demo, we embrace the mindset of creative innovators. We all keep innovating ourselves. In this teaching demo today, I'm going to share with you 2 examples focused on the engineering innovation that may bring us a solution for the current outbreak of the deadly coronavirus. So my understanding of being an engineer, for example, we need creative innovators. We need to keep moving forward to identify the solution, identify the needs of the society and find the best solution and make the world a better place. For the core value, I believe, for my personal belief, I think we need relentless. We need keep fighting and not fear of the failure. Also, we need to be inclusive. We need open-minded, accept the criticism and embrace diversity. Others, we need to have the conscience. For example, we need to be responsible for what we have made, whatever we have made, the bridge or the chemical factory or the airplanes, or maybe the pressurized vessel in the factory, or biocontainment. There's a conspiracy theory. Some people believe this virus was designed in a lab. But the biosafety, biocontainment, as a bioengineer, I think is the top priority to consider. Also, we need to be creative. We need to keep innovating and go beyond the existing solution space. Other thing, we need to be passionate. We need to keep learning new knowledge, inquisitive, and add new skills to our capacity toolkits. So I like this design, build, test, learn cycle that exactly tells how the mindset of a chemical engineer or bioengineer should look like. So let's talk about the COVID-19. This global pandemic has caused hundreds of thousand people deaths right now. So we know this coronavirus. I'll just try to go to the pointer mode. Okay. Okay, I'll go to the pointer mode. Pointer, just with 1 minute, okay. This is a point, okay. So we know the coronavirus actually made with a single strand RNA. This has the spike protein, amyloid protein, some of the glycoprotein. We can build, this is the antigen. We can build antibodies from this, antigen. Also, they have the capsule nucleocapsid capsid protein. So it specifically bound to the human ACE2 receptor protein. So when people catch the coronavirus, they have a fever, fatigue, they have a major lung dysfunction, infection in the lung, major failure in the kidney, and even some other circulatory system. So what's the challenge that this global pandemic, the onset, the progress of this disease has a different character. As the initial 7 to 21 days, 1 week to 3 weeks, we found that we can only detect the viral RNA and the antigen as the cells start entering the human body. But after around 7 to 10 days, the IgM antibodies start become emerging, we can detectable. And after 2 weeks, the cells start making the IgG. So most of the people died actually at this region from the 10 to 14 days after infection for some immune system compromised people because the patients haven't been able to catch up the antibody, build up the antibody. This global pandemic actually requires the developing point of care swab test outside of the hospital and clinical labs because we don't have the hospital result everywhere. So people with suspect symptoms, they can quickly access, they can find out if they have this coronavirus. So at different biological markers, we have different clinical significance. For example, with the RT-PCR, we can directly detect the viral RNA. For the antibody test, we can IgM, IgG, we can do the immunological test. So basically have different clinical significance. So the first thing I want to mention that the first generation of the coronavirus kits, detection kit based on qRT-PCR, which is the quantitative reverse transcriptase PCR. We know all the PCR has 3 steps: denaturation, annealing, and extension. But this PCR is very interesting after we capture the viral RNA from a swab from the nose or from the throat. So we can do a reverse transcriptase, we get the cDNA. During the replication process, polymerase reaction process, we have a specific DNA probe. This probe contains fluorophore and also a quencher molecule, which as the polymerase start proceeding to make the more DNAs. So the molecule actually, they will cleavage this quencher molecule at each of the round of the end of the strand completion of the synthesis. So the nucleus DNase activity of the polymerase, they will cut off the bonds so they can, fluorophore will be released, they can emit the fluorescent signal. That's why we can track each step of the polymerization, the extension process. Of course, we need a very expensive experimental setting, a very expensive chemical agents, or the DNA or probes. We also need the qRT-PCR. We also need a well-trained personnel to analyze the data. For example, we use the CT value, which basically the threshold cycling cycle to determine the abundance of the initial viral RNA. But so this has great limitations. I have friends working in Beijing, it's just a biological lab, but all their qRT-PCR machine was called up to send to ship to Wuhan because at that time in January, there's not enough of the qPCR kits to detect the virus. So because of this time-consuming, tedious process, also they need a specific training. The sensitivity is okay. They can up to 10 to the minus 12, 10 to the minus 9 molar of the viral RNA. But because of the tedious process, also loss of the highly trained personnel, they always introduce loss of the high false positive results. So one of the innovation actually developed in MIT in Feng Zhang's lab, this couple years ago, actually, before the outbreak of the coronavirus, they have this Sherlock, basically depending on the Cas13 CRISPR nucleus. So this is a protein, specifically they can use the guide RNA, they can specifically cleave the RNA, they use the collateral RNase activity. So once the guide RNA, you can design guide RNA on the recognize some of the viral components. Once you have this recognized, the cutting side of the Cas13, they will have a collateral RNase activity. They can specifically cut the fluorophore and the quencher molecule. They can introduce, they can give you the fluorescent signal. This is a flow chart. Basically, you have a double-strand DNA or maybe the RNA virus. You can do a specific amplification process. So you can, at room temperature, you do not need a thermocycler. You can get lots of the cDNA. Once you have the cDNA, you can do a T7 in vitro transcription on the single test tube. You can get RNA. Even later, you can just test based on the collateral RNA activity. So because the thermocycler is a great limitation, actually, people have been invented another technology called RPA. It's called recombinase polymerase application. This application process, they can just take place at room temperature. For example, they only need the mix of 3 enzymes, the DNA polymerase single-strand DNA binding protein. Also, they need the recombinase. So once the recombinase bound the primer, they can start a single-strand displacement reaction. So you can keep amplifying the DNA without the denaturation and annealing and extension process. They only do annealing and automatic extension. So we would say the Sherlock, they combine the simplicity of the recombinase polymerase reaction and also the high sensitivity of CRISPR technology. So, for this problem, I think that one of the essential things, first way to identify the challenge because of the point of care, RT-PCR cannot meet the demand. So this Sherlock actually, the high sensitivity can detect RNA virus up to attomolar. It's 10 to the minus 18 molar. It's very specific. Compared with Cas12, it can also detect DNA, double-strand DNA or maybe DNA virus, but they do not need the PAM sequence. The Cas13, they do not need the PAM, which can greatly unleash the application potential. Yes. So another thing that how about we have multiple viral input signal? For example, you have influenza, we have Mobile virus, or maybe we have other Zika virus. If people catch multiple virus, or maybe coronavirus, also one of the things that orthogonality issues. So a different company right now started working on this innovation. They start working on the Toho switch. So the Toho switch basically is a DNA, is an RNA molecule. This RNA can fall to a special 3-dimensional structure. They can sequestrate the ribosome binding site and the AOG, the translation initiation site, the start codon. So only with the presence of viral RNA. So the viral RNA, they start complement with the Toho structure here, the Toho structure. Once they starting the base pairing, the complementary process, they can displacing, they can open the switch structure. So we can export the ribosome binding site and AOG start codon. So the ribosome can join in, so the cell can make a reporter gene. So this is the flow chart, how you design the Toho switch. It's a single, so if you immobilize this Toho switch, the specific polymerase ribosome on a test tube, on the test strip, you can develop a point of care coronavirus. The beauty of this technique is that you can link multiple Toho switch, you can do orthogonal tests because the flexibility of the RNA structure allows you to design lots of the different Tohos. They sequentially connect with each other. For example, each of the Toho has a different unique viral recognition sequence. Also, they have which can base pair with the viral DNA. They have a ribosome binding site, translation starting site. You can have a different reporter gene. If the Zika virus binds to the Toho switch, they start, unfold the Toho structure. So as a thermodynamic competing process, so the Toho will unfold, eventually export the translational starting site. So the ribosome, they can start making the reporter gene. Normally it's a pink protein or the fluorescent protein or some other protein. It could be very quick because translation can be happening in a few seconds or minutes, a few minutes. The Toho switch, the thermodynamic folding, unfolding can happen in a few milliseconds. So this can just allow you to detect lots of virus. Of course, we cannot just rely on the genetic test. The very important, we also need to look at the immunological test. On the COVID-19, people can develop this cheap, basically the immunological test. On the natural cellulose membrane, we can just have this, we flush through our samples, which contains our antibody, COVID-19 IgM, IgG antibody. After they mix with the conjugation pad, which containing the COVID-19 antigen, so they will form a complex. So once they form the complex, they will form the specific the IgM antibody and IgG antibody. We have M-land and G-land. Of course, we need a positive control. The positive control, we use rapid IgG conjugate. So the positive control can only bound to the anti-rapid IgG antibody. So the positive control basically tells you whether the tests are success or not. If the control doesn't show up, it's a positive control doesn't, it means the tail failed. You cannot interpret any results. Because based on the antibody, so the good thing about antibody, because for transportation, for sample-wide, the antibodies are very stable compared with RNA. Also, which makes them suitable for long-term storage and transportation. We can also, the antibody can reduce the chances of the false negative results. So the data can also be used to interpret the past infection history. So that will be a very good point of care test, just like HIV, HBV test. So the next part, next half 15 minutes, I want to give you another example. Imagine you have an engineered CHO cell or yeast cell that can specifically produce antiviral therapeutic vaccine. So how do we quantitatively understand and optimize the vaccine production process? So it's about fundamentals of bioprocess engineering. If we talk about bioprocess modeling, the first important thing we understand how the cell grows in the bioreactor. So we have this is the simple equation with the substrate, the cell, you have product and more cells. Basically, the substrate with the cell, you have some product and more cell. We have this intraspecific constant we call specific growth rate. So it just describes the fitness of the growth potential of the cell. It's defined with the rate change of the cell density. This is the growth rate, dx over dt, basically the growth rate. After we have this one, it can be normalized by the initial cell density we call specific growth rate. So the reason we introduce this specific growth rate, it's very different from chemical engineering. We just use the rate or rate constant, which is molar per liter per hour, gram per liter per hour. But here, the reason we introduce specific growth rate because the cell living system, they keep proliferating, they keep growing themselves. That's a self-catalytic reaction. So we need to normalize cell density to give us a time scale. So the specific growth rate will be the per time. Imagine if you have exponential growth, which means the specific growth rate, the constant value, we can just do an integration. We found the cell density, it just exponentially grows with time. From this exponential growth, we can define another term we call doubling time. Doubling time, which is the time spent for the cell to double their biomass. We found that the specific growth rate and doubling time has an inverse correlation. It's ln 2. It's easily detected. I want to mention, this is very critical for being a chemical engineer or biochemical engineer. So lots of time people doing experiments, they found actually the specific growth rate is depends on the substrate concentration. They follow the hyperbolic equation we call the monod growth. It's well fitted to a monod growth curve. For example, the specific growth rate mu is depending on this hyperbolic function. So mu max is the maximum specific growth rate, and the Ks actually is the saturation constant. It tells us the preference of the substrate. If the Ks value is low, it means the cell only needs a tiny amount of substrate can become saturated, which means this substrate is very suitable or maybe very yummy for the organism to taste it. But if you have a very high Ks value, which means the cell doesn't doesn't lack this substrate. We found actually this is the mixed kinetics. At the first stage, if the substrate concentration is well below the saturation constant, we found it's approximately a linear growth curve. It's a linear dependent relationship. But if you move well beyond the Ks value, we found it's go to a zero kinetics. So the mu almost approaching to the mu max. But at the first stage, at the well below the Ks is the first order reaction. So it's the mixed order. Eventually, we can say this is the mixed order growth curve. So with this growth curve, I want to demonstrate how we can do a mass balance in the bioreactor. Probably here, I just plug in one thing. I can show some of the, okay, I can show some of the things here. So, okay, you can show here, if we do the mass balance here, we imagine we grow our cell in the CSTR, and the biomass is the only product. So CSTR basically the continuous stereotype reactor. We have a constant flow goes in the bioreactor, constant flow goes out of the bioreactor. Also, we have initial substrate concentration, initial cell concentration. We'll assume it's zero. We assume there's already has some initial cell in the bioreactor. So we have well mixed. There's no mass transfer limitations. So based on this, CSTR, we can first define the mass balance for cell growth. V times dx over dt. Basically, that's the units for gram per hour. It's the mass of the cell growth per time, because dx dt, the rx is the cell growth rate. So we have the mass balance is equal to the mass of the cell goes into the bioreactor is equal to F times the x naught, and minus the mass of the cell leaves the bioreactor or carry out from the bioreactor. Also, because the cells are living system, they keep growing. Also, we should consider about the V times the Rx. Rx the cell growth rate. If we plug in the Rx equal to Vx, at steady state, we assume that the dx over dt, of course, steady state, dx, there's no cell concentration change. Dx over dt will be zero. We found because initial cell concentration is zero, we can just initial cell concentration is zero. So I'll just change my pointer to a pen. I think I can do some maybe annotation on the. Okay, I change my pointer to a pen so I can do the annotation for the. Okay, so okay, yeah, the annotation, okay, it looks the annotation is quite, just wait, okay. Okay, so yeah, I'll go back to make the pointer because the annotation didn't work here. Yeah, excuse me, yeah, some technical issues here. Okay, so basically the x naught could be zero. Once you have the zero, you can find the next equation here. So the fx will be, you will find the mu actually can be, the x can be canceled out. You get mu equals d. Eventually, because we know the cell follows the monod growth, inversely, you can solve the substrate concentration here. It's about KSD minus mu max minus d here. I still try to find my pointer here because it goes to a special mode here actually. Okay, so yes, okay. I'll just remove this one. Okay, I just remove the pointer here. I just go to the pointer. I'll still go to the pointer mode because it makes it more easy for us to follow up. Okay, so we can also do the mass balance for the substrate balance. We have it's a little complicated because the substrate was initially fed into the system. So before we do the substrate balance, we want to define a gross yield. We want to correlate everything the substrate consumption with cell growth because the most distinct character of a bioreactor is the cell growth. So we define the gross yield. It means every unit gram of the substrate consumption, how much of the cell you can get. Basically, we found there's a correlation. The cell growth rate and the substrate consumption, there is a correlation called y-axis. It's a U coefficient. What happens is that when you do the mass balance, so we have V ds over dt. You found this is the substrate consumption rate net a change of the substrate. It can equal the substrate goes into the bioreactor and the substrate goes out of the bioreactor. Of course, the substrate consumption rate. The substrate consumption with RS can be plugged in because we already know the RS actually can be plugged in the mu max divided by y-axis. Eventually, you solve this equation, you found x equals this one. You plug in the S concentration, steady state substrate concentration, you will get X equal this. So this is the steady state cell concentration. But I want to ask you one question. If you plot this solution, steady state substrate concentration and steady state cell concentration as a function of the dilution rate. So you found actually the cell as you increase dilution rate, we found the substrate concentration keep increasing because the utilization efficiency is slow, but the cell concentration also starts decreasing. Actually, we found if we imagine the cell biomass is the only product, we found that there is a Rx or the cell productivity. Actually, there is a plateau value, there's a peak value. So we want to know what's the optimal dilution rate to reach the maximum productivity. So the optimal dilution, the productivity can be is a P equals Rx, which is dx. From this equation, we know it's depending on, there's a linear order, first order on D, there's another parabolic on D, which once we do a dP over dd, it's a capital D equals 0, we will find the dilution rate actually is around this formula. So basically, it's an easy process. We can predict under which dilution rate we can maximize the productivity. But this is just one thing. A lot of the time, the product are not just the biomass. We have a real product. In the real biological system, the product might have 3 different distinct patterns. We call growth associated. Those, this, for example, here, the growth associated, the product formation is well associated with the growth. And also we have partially growth associated. The product P is just partially associated with the cell, or maybe the unassociated model. The cell didn't make product at the beginning, only the cell enters stationary phase, the product starts coming out. So people found that we can define a correlation we call QP, the specific product formation rate, is linearly correlated with the mu, specific growth rate. We use the constant term alpha, mu, and beta. So alpha is the growth associated parameter, beta is the non-growth associated parameter. If alpha equals 0, it will become a completely non-growth associated model. If beta equals 0, it will become a completely non-growth associated model. So we also try to introduce another concept called product yield because we know everything, the growth rate, or production formation rate, we all want to link up to the cell growth rate. So based on this rate, we can divide it by delta T on both the delta T and delta S. On the top, we will get RP, which is the product formation rate. On the bottom here, we'll get RS, the substrate consumption rate. We know the substrate consumption rate can be defined with the QP times X, the specific product formation rate. So we can plug in the alpha, mu plus beta. Eventually, we found the substrate consumption rate. The reason we do this is because the substrate consumption was used not only for biomass, also for product. For example, the change of biomass in this scenario will include the substrate carry in, carry out due to the feeding, also include the consumption of the substrate because of biomass, also including the consumption of biomass, consumption of substrate because of product formation. Eventually, we can list the mass balance equation. This is the monomer growth. This is the cell growth equation. This is the mass balance for the substrate consumption. So this is a carrying out term. Here is the growth term, substrate consumption because of cell growth. Here is the substrate consumption because of product formation. The last equation will be the product formation mass balance. Of course, at the steady state, we can also reach the solution. It's easy. My student, I can ask my student, they can do Excel. So this is corresponding to a partially grossed model. So the P is corresponding to the product, X is the cell biomass. So RP is the product productivity, and the Rx is the biomass productivity, and the S is the substrate consumption. Of course, we can change the alpha beta value to understand at which condition we can also arrive at an analytical solution, at which operational dilution rate we can achieve the maximum vaccine productivity. Of course, if you further increase dilution rate, you get a washout. You won't be able to produce any pharmaceutical. So the last thing I want to mention basically is what's the innovation concept coming? Because lots of the time when you produce a vaccine antibody, you use a very expensive media. Use FBS media with bovine, with the blood syrup. It's very expensive. Also, you want to, you don't want to just discharge the media and just do another batch because the costs are very expensive. Actually, people else sometimes the vaccine production, the productivity is very low because it was specific yield. People have been in the process engineering process. They can design a specific cell retention device. Basically, they can do a cell. They can pump back some of the concentrated cell back to the bioreactor. And they harvest some of the broth. The broth contains some of the cell and contains some of the vaccine, the final product. But the concentrated cell, which is the factory for the vaccine, it can go back to the bioreactor. So we can keep the cell concentration here in the normal steady state vessel. For example, here in the normal chemical plants, this is the process. You have this perfusion culture. You have this cell retention device. You can go back to the perfusion rack. Eventually, you can also harvest the clarified harvest. If we compare the efficiency, of course, we can do mass balance. We have, if we set up the recycle ratio, the concentration factor, we found actually the simulation actually is the analytical solution. If just with the CSTR, no cell recycle, the productivity here, so P1 with the blue curve with the productivity of the vaccine is very low. But if we can move to the perfusion bioreactor, it's a purple curve here, it's a P2 we found, it's much here. That's why we can increase the container utilization efficiency because the pharmaceutical industry, the capital investment is very high. Also, we can minimize the media cost because we can. That's why we can increase almost 90% of the pharmaceutical process use perfusion bioreactor now in the industry. So, at the end, I want to give my students a few inspirational homework. Can you derive the steady state solution for the logistic model and hybrid logistic model? For example, logistic model give you the boundaries which tells you the cell growth was inhibited by the biomass. Initially, actually, if you do a separation variable, you can manually derive the integrated equation. But you can, if you plot the equation, change the mu max on the MATLAB, you can find cell density. How does the cell density change with respect to time? Another thing I just mentioned in my research seminar, actually with the hybrid logistic model cell growth model, you may be able to capture both the nutrient limiting condition and the growth limiting condition. So we can derive, I would encourage my students derive the analytical solution in both the batch culture and CSTR culture. So about the RNA structure, so actually reading, So on aptamer for COVID-19 detection, we know aptamer structure. How can we design or evolve, engineer a specific RNA molecule with specific ligand binding affinity? There's a very cool technique called SLEX. Basically, that's called a systematic evolution of ligands by exponential enrichment. If you can, based on the pooled mutant library on the specific antigen antibody, you can design and fishing out selectively enrich the RNA sequence that's with increased binding affinity. You can keep running this cycle. Eventually, you can find this capture aptamer or detection aptamer. So the RNA is the aptamer. Eventually, you can immobilize the aptamer into a natural cellulose membrane. You can capture the protein. On the other side of the aptamer, you can attach to a biotin or streptavidin binding domain with the enzyme. So enzyme can give you reporter gene or maybe give you light or pigments. Of course, you can design a sandwich-based structure. You can have the target protein in the medium. You can have a capture aptamer and a detection aptamer with the reporter gene. So this could be another cool technology for detecting COVID-19. So my overall teaching goal, basically the course I'm interested in teaching some reaction kinetics, chemical engineering, metabolic engineering, synthetic biology, or biomolecular engineering. And the course I want to develop including numerical simulation in bioengineering. I try to introduce both deterministic or stochastic approach, or quantitative biology, computational system biology, biointelligent control and design. So the teaching goals I would like to attain, including I want my students to be critical thinking, creative, they can acquire computational skills, they are good at communications, also they are good at teamwork, they can collaborate. I think the creativity, can we teach the creativity? I think it's more about the environment. It's about the thinking process. So once the student has the domain knowledge or the knowledge structure, these are the foundation for the creative. Also, we need an open environment. We need to encourage the student. We can use gender neutral language. We always encourage, never criticize, inclusive action. We encourage everyone to participate. That's why it's so important to promote diversity. We can show our respect to every student. We want to make students are active learners. They want to know why it's so important to learn this technique, where they can find a job, why it can benefit the society and the human, how it can make economic or maybe society impact. Also, of course, we can do reflective teaching. We want to make connection with our existing knowledge, our daily life. Also, it's very important. I want to do a measurement and assessment and midterm feedback evaluation and keep improving the teaching process. Okay, thank you for your time. At the end, I want to thank you for the time. I will be open for questions.
Yep. Do we have questions from students, firstly, or pseudo students?
Sure. Okay.
Well, it appears that that was a long lecture, and I wonder how many people are still left here. So the second call for the questions, the clarifications.
Okay, it's on the chat. It's on the chat, or it's on the.
Well, for the moment, we do not have questions.
Sure. Okay.
All right then. So if there are no other questions, I would, in the interest of time, I think Peng would need to meet other people anytime soon. I will close the presentations now, and you should proceed with your timetable for today. Thank you, Peng.
Thank you.